And the last thing, and this is kind of one of the most interesting parts of meiosis, is that there's a weird process called homologous recombination that takes your mom and dad's DNA of a given chromosome and mixes it all up. And that's something that doesn't happen during mitosis, this process of homologous recombination. So in homologous recombination, you actually mix mom and dad's homologous chromosomes. So if you keep these things in mind, all right, that it's a type of division that only occurs to give rise to eggs and sperm, you have the amount of DNA. At the end, regardless of how many chromosomes you're talking about, you always make 4 cells. And you recombine the homologous chromosomes during the process, you kind of have the big picture of what meiosis is. You have a question?
Yeah, so is that actually, remember there was, like, we were talking about all the ways that the DNA could be recombined, and we were like, oh, well, you know, it could be this, or it could be this ridiculous thing that we're not talking about. Is that actually, like, kind of what that is? That this is the, well, I forgot the name of it.
Dispersive.
Dispersive, yeah, is this?
Yeah, I'm hesitating because I've seen some images of dispersive replication where it's a single chromosome that's double helices that are either old or new, sort of interspersed with each other.
Oh, okay.
So in that sense, there is a similarity, all right? But in this case, homologous recombination doesn't involve replication. It occurs well after replication, all right? So you replicate the homologous chromosomes, you make the sister chromatids, and then the homologous chromosomes swap DNA. Like, it's not a process of copying, it's literally a swapping of information between them.
Although, so it looks kind of similar as an end result, but it's not a...
There's some similarities to the fact that it's sort of cut and spliced together,
yeah.
But it is a different process, yeah. And it's not confirmation that it's dispersive and not semi-conservative.
Yeah, okay.
Okay, so these are kind of the things to keep in mind. Reduce from diploid to haploid, make 4 cells instead of 2, and recombine the genetic information. Okay, so like last time, I'm going to sketch it out for a simple situation, a single cell with 2n equals 2. I'm going to walk through the whole process of meiosis, and by then, you guys will be ready for a break. I'll be ready for a break. We'll take a break, and then we'll start the lab. And you guys will spend, probably, some of you probably have some work left to do on the mitosis drawings. So you can work on the mitosis, and then we'll work on the meiosis as well. And then we'll kind of walk through at a certain point and make sure everybody gets the big picture of how meiosis occurs. So